How do we see? How does the human eye function? This video is going to discuss the visual path, tracing all the steps required for humans to participate in the spectacle of conscious, visual experience. So there are three main phases of the visual path. There is the path of light. How does light get into the eye? What changes occur as it enters? And what structures in the eye help to facilitate this entry? The second phase deals with all of the electrical changes that happen at the level of the photoreceptor and its ancillary structures within the retina. What is a photoreceptor? How does it transduce information? And then there are the resulting nerve impulses that are sent from the retina of the eye to the visual processing module of the brain. How is the signal wired and routed? Where is it ultimately processed? Light, retina, brain. Let's start with light, which is the information in the external world that your eye is interpreting. Light first enters the eye by passing through the transparent cornea. The convex shape of the cornea refracts the light, converging the rays through the pupil to a more centralized focal point on the lens. The lens refracts the light even more. This allows the rays to enter the retina at a common focal point. In the retina, the light continues to gleam through a forest of ancillary wiring before finally reaching the transducer component of the visual system, the photoreceptor. A photoreceptor is a cellular machine that transduces certain wavelengths of light into electrical impulses. These impulses are then sent along a system of interconnected cables that extend from the retina of the eye to the visual processing module of the brain. A photoreceptor has three main parts. The inner compartment contains all the normal cellular essentials, like DNA, mitochondria, and other important organelles. There is a connector component, or synaptic terminus, which interacts with the adjacent cables. And there is an outer segment, which is the actual transducer component. The specific molecule inside the transducer that reacts to light is a derivative of vitamin A known as retinal. Retinal is bound inside a protein called opsin. The two molecules are collectively referred to as rhodopsin. As light beams through a photoreceptor, inside the transducer component, it splits rhodopsin into retinal and opsin. As the photons surpass the photoreceptor, they beam right into pigmented epithelial cells, where they are absorbed by granules of melanin. This is the end of the line for the path of light, but with rhodopsin split, we now have an activated photoreceptor. So let's take a deeper look at all of the changes that occur inside this remarkable cellular apparatus. Keep in mind, the chemical events we are about to discuss occur very rapidly, that is, in a matter of femtoseconds, or quadrillionths of a second. Note, the means by which photoreceptors convert light into electrical signals is a process known as phototransduction. As mentioned earlier, the process of phototransduction officially begins when light makes contact with rhodopsin, splitting it into retinal and opsin. When rhodopsin splits, this activates the G protein, transducin. Transducin activates the enzyme phosphodiesterase, also called PDE. Once activated, PDE begins hunting for a specific nucleotide inside the photoreceptor, known as cyclic GMP. Okay, a quick aside. Cyclic GMP is a nucleotide that lives in the cytoplasm of virtually all human cells. It primarily functions as a messenger molecule to relay signals in many transduction pathways, like the phototransduction pathway. In the dark, that is, before light enters the photoreceptor and splits rhodopsin, levels of cyclic GMP are high. When cyclic GMP is in abundance, it binds to receptors on sodium channels anchored to the cell membrane of the photoreceptor. This keeps the sodium channels open, which allows sodium ions to diffuse into the photoreceptor from the extracellular fluid. The sodium influx causes the photoreceptor cell membrane to remain depolarized, which in turn keeps voltage-gated calcium channels at the synaptic terminus open. This results in an influx of calcium ions into the synaptic terminus from the ECF, which triggers the release of the neurotransmitter, glutamate. Glutamate diffuses across the synaptic cleft and binds with chemically gated ion channels located on the telodendria of an adjacent bipolar cell. These inputs trigger shifts in ion concentrations between the bipolar cell and its extracellular environment, such that the cell's membrane is continuously hyperpolarized, i.e., it keeps it in the off position. However, when activated by light, PDE hydrolyzes cyclic GMP, causing it to unbind from the sodium channels. When cyclic GMP is released from the sodium channel receptors, these channels close and sodium influx is halted. But because potassium continues to leak out into the extracellular fluid via potassium leak channels that remain open at all times, the cell membrane undergoes hyperpolarization. The hyperpolarizing current then propagates down the photoreceptor membrane to the synaptic terminus, where it triggers the voltage-gated calcium channels that were previously open to close. This halts calcium ion influx and ultimately stops exocytosis of glutamate across the synaptic cleft. The bipolar cell across the synaptic cleft no longer receives neurotransmitter inputs from the photoreceptor, which releases it from inhibition, and it undergoes depolarization. In truth, it's a bit more sophisticated. Indeed, some bipolar cells do, in fact, depolarize when released from inhibition. We call these bipolar cells on cells. Other bipolar cells do the opposite. That is, they hyperpolarize when released from inhibition. And we call these varieties off cells. This enhances contrast in the visual field. The impulse is then transmitted to a ganglion cell. If the impulses generated in the ganglion cell body combine to reach threshold, an action potential will fire down the ganglion cell axon. There are around 1 million ganglion cells in each retina. All of the ganglion cell axons coalesce into a single cable known as the optic nerve. Okay, it's finally time for us to leave the retina and head to the brain. So, how do we make this move? The optic nerve extends in a posterior direction from the eye. Some axons originate from the medial section of the eye. Others originate from the lateral section. The optic nerves of each eye intersect at a point within the cranial cavity known as the optic chiasma. From here, the axons of the medial retinas of each eye cross over and wire into the opposite hemispheres. The axons from the lateral retinas, however, do not cross over. Instead, they wire into the hemisphere on the same side. Once we surpass the optic chiasma, the axons are no longer referred to as optic nerves. Instead, they are called optic tracts. After crossing over, these tracts extend in a posterior direction and synapse with neurons on a ralter in the very center of the brain known as the thalamus. From the thalamus, multiple impulses are then transmitted along white matter tracts to the visual cortex to be integrated and processed as conscious visual experience.